Okay, in this video we are going to be looking at the next step in our course, which is muscle contraction. And the reason I place this video and this lecture in this position is because we have been examining ATP generation using the pathway of glycolysis in red blood cells. Now recall, from glycolysis in red blood cells, this is going to be generating a simple 2 ATP per molecule of glucose. Now, this is a very inefficient method of generating energy from the oxidation of glucose, but glycolysis is simply sufficient and necessary for red blood cells because the energy requirements of red blood cells are rather minimal. The next phases in oxidation of glucose beyond glycolysis will include the TCA cycle and will include oxidative phosphorylation. These processes don't occur in red blood cells because red blood cells are the carriers of oxygen bound to hemoglobin to deliver into the tissues. Therefore, oxidative phosphorylation, the final electron acceptor being oxygen, will not occur in red blood cells because red blood cells have a different function. They need to carry oxygen rather than use the oxygen to be an electron acceptor. Now, this being the case, what we need to examine is the ATP requirements in muscle. And I place it in this context so that you can make a comparison to the amount of ATP needed for red blood cells and that needed for muscle. So ATP, again, in red blood cells is a minimal need. Now, for muscle contraction takes a lot of ATP. Let's examine the different properties of the structure of the contractile element and see where the ATP is used. So the first protein we have to look at is called myosin. Myosin consists of two large subunits, subunits being individual polypeptides, and in combination with four small subunits, four small polypeptides. So when we talk about myosin, myosin is actually a complex. It's a complex of 6 polypeptides that come together. Now, the structure of myosin is essential for the function of myosin. So let's take a look at the structure. I'm going to draw myosin, for example. The first large subunit is a snake. The next subunit will be coiled around the first subunit. So this portion of myosin represents the C-terminal portion of the large polypeptide subunits, and this is called the tail of myosin. The tail of myosin will, as we see in a minute, will perform the interactions to form the large myosin structure in muscle. Now, toward the N-terminus of myosin, we have two globular domains, and these globular domains are referred to as the head groups of myosin, of which there are two. Now, the smaller subunits that are part of the myosin complex associate with this portion of the myosin. Important, the tail interacts with other myosins. The head of myosin will interact with actin. Now let's look at the second major protein of the contractile element, which is actin. Actin is a single polypeptide. It is a single subunit protein. and it is a globular structure. Now, the important definition of globular in this case is that the free actin that is soluble in the cytoplasm, that is not bound into the filament structure of actin, is termed G-actin, for globular actin. The G-actin then will undergo a polymerization reaction to form F-actin. The F in this case stands for filament or filamentous actin. And this simply indicates that the globular units have come together to form a filament. Now, in this process, the G-actin itself will bind ATP. So here's one of our first steps of using ATP in muscle, is just establishing the contractile element. So G-actin binds ATP. The ATP is hydrolyzed, releasing the inorganic phosphate. The F-actin maintains the binding to the ADP. Now let's take a closer look at the structure of how these subunits come together so that they can perform their interaction. The myosin, for starters, has this tail region. Let's depict the tail as this bar, and we have head groups coming out of the N-terminal portion of the complex. Because these tails are able to interact, what the myosin is able to do then is form a long filament by repeated binding of the tail portion of the myosin molecule. This is one direction, but we can flip this and we can form these tail-to-tail associations in the opposite direction. And again, with our head groups coming out of the N-terminus. So this is essentially the complex just flipped in the opposite direction. Now, because of these associations, these repeated associations of these tail groups, this will form what is called a thick filament. Now, the term thick filament comes from electron microscopy. So if we look at this structure in an electron microscope, this will seem rather thick, which means a dense view to it. Dense equals dark on the picture. In order to generate this electron dense material, appearing dark, you need to have lots of protein. So when lots of protein come together on an electron micrograph, this will appear dark, hence the name thick filament. Now the actin itself will form another filament. When the G-actin comes together and forms the F-actin, these globular units of actin will form a long filamentous structure, and it will appear that these form a coil, the actin. So the F-actin forms these long strands. Now, these are globular protein units that are only coiled in this dimension, and they are not stacked on top of each other like the tails of myosin. So this, in an electron microscope, will not look quite as dense because the density of the protein is not as much. These are termed thin filaments. Now, in order to make the contractile unit, we need to have an interaction between the myosin, thick filament, and the actin, thin filament. So the way we're going to do that is we are going to put the actin in this dimension. So the actin will extend and overlap the myosin filaments. In this way, we have the potential for interaction between the thin filaments and thick filaments. So what we'll see in a minute is that these heads will interact with the actin, and because of the opposing direction of the thick filament of myosin complexes, these will pull the thin filaments in toward each other. Now, in order to get contraction, we have to have something tethering and holding these ends. What we have is a bundle of proteins that are anchored to connect these thin filaments. So the thin filaments are tethered at the end. So when the actin performs its interactions with, myosin performs interactions with actin, it's going to be pulling on these bands. Now these bands have names in the electron microscope. We have actin filaments coming out the opposite direction, of course, like this. And this particular region is going to be very light with the central band, and this is called the I band. The central portion, where it's predominantly thick filaments, will be more electron dense. This region is called the A band. So the I band pretty much corresponds to the F-actin thin filaments. The A band is the myosin complexes, which is the thick filaments, thin and thick, giving the different electron densities. Now, this entire unit is repeated over and over and over again millions of times and duplicated in order to form fibers. Smaller ones are called fibrils, and these are bundles of individual units called sarcomeres. So when we move from the dense band, I band region, to the next one, this essentially is a contractile unit, and this is called a sarcomere. So the sarcomere becomes the primary unit of contraction. Now let's look at how contraction actually occurs. What we'll take a look at now is the biochemistry of contraction. It's at this place, this stage in contraction, is where the majority of the ATP in muscle contraction is going to be used. So if we have our thin filament of actin, so this is our F-actin polymer. We have myosin interacting with the individual heads of the actin. So let's draw 3 actin monomers, and we will draw a myosin head interacting with the actin and then the tail of the myosin. Now, in order to perform contraction, what is going to happen is that this myosin head is going to ratchet. It is going to dissociate from the actin, bend, reassociate, move. And in these form, in this repetitive action, we'll be able to pull the thin filament. So how does this occur? First thing we need to do is introduce ATP. ATP will bind the myosin head group. And what we will have in this position, I'll just draw the 3 actin filaments that continue on the filament. We will have now the head group dissociated from the actin monomer after it binds ATP. So ATP induces the dissociation of actin and myosin. The next step of the reaction is the hydrolysis of ATP. The hydrolysis of ATP will release inorganic phosphate. What this essentially does is it causes a conformational change in the myosin head group. The ADP stays bound, remains bound to the myosin head. But notice we have a conformational change. The way to look at this is that the hydrolysis of ATP is going to be releasing free energy. This negative delta G, this free energy, is going to be captured in the conformational change, the movement of the head group. So picture this as a spring. This is a relaxed spring. With the transfer of this free energy from ATP into the myosin head group, picture this now as a tense spring. So we are transferring this free energy that we gain from ATP hydrolysis into potential energy by this conformational change. So this conformational change is holding the energy of this release from ATP hydrolysis. The next step now is with ATP bound to the head group. The head group now will associate with the actin subunit. But notice the actin subunit that it is interacting with. Originally it interacted with, say, number 2, and now that we have ratcheted the head, it is now interacting with actin unit number 3. The final activity here is called the power stroke. What the power stroke simply is that we are going to release ADP from the myosin head group, and when this happens, the stored energy, because of this conformational change, the energy gained from this free energy change of ATP hydrolysis, we are now going to release this energy. And the releasing of this energy is going to be done in the form of work. The filament will now be pulled. And the actin head will remain bound, but return to the same position it started, except it's now moved 1 actin subunit down. Now, regulation. Of course, we want our actin-myosin interactions regulated so that contraction is a controlled, smooth event. And so what we are going to have here is neuroconduction. This, of course, is coming from our nervous system, come from our central nervous system and our peripheral nervous system. And we have a wave of impulse, nerve impulse, propagating down the nerve to the nerve terminus. This is now considered a neuromuscular junction at the end of the nerve synapse. This synapse now interacts with the muscle cell. It interacts in a way that acetylcholine is released from the nerve terminus due to this impulse. So this electrical impulse, depolarization, induces these vesicles at the nerve terminus to fuse with the membrane and release acetylcholine. Acetylcholine is a neurotransmitter. Its job is solely to transmit the nerve impulse, electrical impulse, depolarization wave, into a chemical signal. This now binds to the muscle, binds to specific acetylcholine receptors on the muscle, and this will now induce a depolarization event in an intracellular vesicle so that calcium is released. This intracellular compartment inside of muscles is called the sarcoplasmic reticulum. The sarcoplasmic reticulum is a membrane-bound vesicle inside of muscle cells, and its specific job is to maintain a high calcium concentration. Upon stimulation of the muscle with acetylcholine, that causes gated channels that are located in the membrane of the sarcoplasmic reticulum membranes to open. What happens then is calcium releases out of these vesicles and now into the cytosol of the muscle cell. The calcium then is going to be the trigger for inducing muscle contraction. And it does so by a very specific mechanism. Let's say this is the thin filament of actin. So this is our F-actin filament. And here is our myosin head. Here's our head, and here's the tail. There are complexes of protein that are bound to the actin filaments. These complexes consist of two specific proteins. One is called troponin. The other is called tropomyosin. And these two proteins bind together, and they bind to actin. When they bind to actin, the interaction of the myosin head with the actin is prevented. If the myosin head cannot interact with the actin, then none of these contractile events can occur. So there's no contraction. What calcium does is when it's released by this nerve impulse, is the calcium now will bind to troponin. Binding of the calcium to troponin now releases this complex from the actin. So plus calcium causes the dissociation of troponin and tropomyosin, and now it allows the actin and myosin to interact. The reverse is true, of course. When the nerve conduction is completed, and we no longer need contraction, the calcium will be recycled back into the sarcoplasmic reticulum by an active transporter. The reduce in calcium concentration now will cause the troponin-tropomyosin complex to reassociate with the actin, thus preventing the myosin from interacting with the actin and ending the contractile event.